little bit.
All right.
Okay,
So
got it.
the screen is yours now. Yeah.
Yes. Okay. Mine is good. Okay, let me just open. 1 second. Okay. So let's start. My name is George Tetz. I'm well, everything that Gabriel. By the way, Gabriel, right? The first name is Gabriel.
No, I prefer with Antonio actually.
Antonio? Antonio. Okay, Antonio. So everything was correct. Graduated from St. Petersburg University. My PhD thesis were specifically also dedicated to the role of extracellular DNA in bacterial biofilms and the role in antibiotic resistance and sensitivity. But indeed, there's a lot of research going on in this field, and I personally agree with Antonio that it's very exciting. We're still to be scratching the dark matter of extracellular nucleic acids, definitely. And so let's start. We started looking at the eDNA or extracellular DNA or cell-free DNA, it's all the synonyms, from a little bit different angle. And I personally will hope that you will enjoy the talk. Okay, so, so today I will be talking about the series of our discoveries. Actually, it's the summary of 3 our papers that bacterial extracellular DNA could modify mammalian proteins in different ways, including cross-seeding, causing a kind of a prion-like aggregation. And I will start with the new role of bacterial DNA as a seeding factor that leads to protein misfolding in Alzheimer's disease. And let me start with a really brief introduction in protein misfolding. I know that most likely it is obvious to everyone who right now is on the call, but just to make sure that we're on the same page. So prion proteins are characterized by cell propagation, undergoing a conformational switch from one state to another, which in turn leads to the creation of a new prions. And pathologically, prions are characterized by a process in which the infections form of prion, which is already misfolded, right? It interacts with indigenous non-prionogenic protein, but it catalyzes its transformation into the misfolded form, with a prion aggregation. So originally, this process was observed only in Creutzfeldt-Jakob disease. However, today it is known as a key pathological process in almost all neurodegenerative diseases, a bunch variety of different autoimmune diseases, and even cancers. Because P53, that is a key for different types of cancers, also undergoes prionogenic misfolding that leads to its inactivation. So examples of the major, and once again, it's not all, just the major protein misfolding diseases and their targets are shown in this table. So one can see that it almost, touches the whole set of multifactorial diseases, which as of today are really, really poorly curable. And it is obvious that the pathogenesis of Alzheimer's disease is characterized by the misfolding of 2 proteins with their prionogenic-like properties. It's beta amyloid and tau protein. Misfolded forms of these proteins are neurotoxic. So their appearance, the appearance of their misfolding aggregates, and their further accumulation in the certain parts of the human brain results in the neuroinflammation, neuronal death, and the disease which is known as Alzheimer's disease. However, the initiating factor that actually leads to the cascade of protein misfolding or the seed underlying prion aggregation remains largely unknown. And just a few words regarding the background of this discovery. Our core was one of the first to show that literally all bacteria and fungi secrete and release cell-free DNA. That is what can be seen on the left-hand side. On the image A. And in some bacterial communities, this DNA was recently shown to convert bacterial alpha amyloid protein into misfolded beta form, also forming so-called curly fibers with prionogenic properties. So please see the image B. In other words, bacterial DNA promotes bacterial protein misfolding. However, nothing was known, literally nothing was known, to be honest, about the cross-kingdom interactions between bacterial DNA and human pranogenic proteins. And now let me switch gears for a moment and discuss the brain microbiome. I know it might sound surprising since brain has been considered historically to be a sterile environment. However, recent data suggest that there are multiple and different types of bacteria and even fungi in the brain tissue of patients with Alzheimer's disease. What's more, recently, some bacteria, including Porphyromonas gingivalis, E. coli, Chlamydia pneumonia, Borrelia species, and Candida, have also been detected in cerebrospinal fluid and postmortem brains of individuals with not only Alzheimer's disease, but other types of neurodegenerative diseases and even health controls. And there are many ways in which bacteria can reach the central nervous system, right? Some through an altered blood-brain barrier from this such as Porphyromonas gingivalis that get directly to the brain by migrating through the olfactory nerve pathway from the oral cavity. And now I would like to talk a little bit about the studies for the role of Porphyromonas gingivalis in Alzheimer's disease and underlying clinical trials. So it was Steve Dominy who originally discovered the Porphyromonas gingivalis presence in the brains of patients with Alzheimer's disease. So Porphyromonas is a type of bacteria that is normally involved in gum disease. Well, it's not normally involved, of course, but it's involved in gum disease. And Steve suggested that it may be the toxins released by these bacteria called their gingivalis that brings on the disease. And in their study, they show that P. gingivalis and their gingivalis, they can fragment tau. And tau fragmentation is known to play a key role in the formation of hyperphosphorylated tau in Alzheimer's disease. So they used their gingivalis inhibitor named COR338 to specifically block gingivalis and to see if it improved symptoms of Alzheimer's disease. Their initial clinical data is positive, and they expect to announce the readout from phase 2, I think phase 2-3, already mid-November this year. So, well, good luck to them. We'll see what it will turn on. However, a few questions remain unanswered. And the first question is, why do some people, even without Alzheimer's disease, have P. gingivalis in their brains? So this is the data from the study by Dominy. So this question was raised by Steve Dominy's team originally, but also unfortunately remains largely unanswered. So you can see that the brains of patients with the control also has, also have the number of P. gingivalis, and even gingivalis are also released inside, from these microorganisms that can be seen on the left-hand side. The second question is that along with P. gingivalis, other bacteria were identified in brains of patients with Alzheimer's disease. The thing is that Dominy originally discovered P. gingivalis in early 2000s, but since then, hundreds of papers have appeared showing the presence of other bacteria, predominantly E. coli or Borrelia and some other bugs, as I mentioned before. And these experiments, just to highlight, all of them are very nicely performed, meaning that, these microbiological signatures are highly unlikely to be, due to a PCR artifact. So let's consider that these studies are done accurately and it's not artifacts. So although the primarily role right now in clinical trials, right, they're put to P. gingivalis, however, that seems to be due to the fact that this microorganism was originally discovered in brains of patients with Alzheimer's disease. However, these studies discount other bacteria and other bugs that were, that are present there. And so just to summarize, right, although there is a lot of research studying the role of the brain microbiome, the pathophysiological processes underlying how brain-localized microorganisms could trigger Alzheimer's disease is still elusive. And we've got a wild idea. We hypothesized the DNA secreted from brain-localized bacteria, as well as bacterial cell-free DNA that can be found in the blood plasma can get to the cerebrospinal fluid from the systemic circulation due to impaired blood-brain barrier and could lead to protein misfolding in the brains of patients with Alzheimer's disease. So therefore, we suggested that bacterial DNA is a promotional or seeding factor for tau and A-beta protein misfolding. We use the protein misfolding cyclic amplification method based on theoflavin fluorescence that mimics in vivo protein misfolding process. And we use the DNA of bacteria previously cultivated directly from the brains of patients with Alzheimer's disease, or identified, other in central nervous system and in plaques or in the circulation or oral cavity of patients with Alzheimer's disease. So we had DNA of Porphyromonas gingivalis, of Pseudomonas aeruginosa, DNA of several, actually of 2 E. coli strains. One was amyloid producing, another not. DNA from Borrelia, and 2 novel species identified by our core from the oral cavity of patients with Alzheimer's disease. As a control, we used eukaryotic DNA of Candida and human DNA. The goal here was to show that bacterial extracellular DNA could significantly accelerate tau aggregation. And this slide actually shows that. So we use the amyloid-specific fluorescent dye, theoflavin T, and its fluorescence, it increases as tau or beta amyloid fibrils aggregate. But this one study is only for tau protein. We will jump to A-beta later on. And each of these lines is the intensity of tau protein misfolding during a certain time period. And the difference between these lines at the end of observation period actually reflects the varying potential of DNA isolated from different organisms on tau protein misfolding compared to the untreated control. The untreated control, you can see it's a purple line. Yep, here we go. It's human DNA. Oh, no, sorry. The untreated control here, it is a blue line right behind of that, and human DNA is a positive control. Sorry about that. So these data show that DNA from various unrelated gram-positive and gram-negative bacteria significantly accelerated tau aggregation. And one of the best promoters was DNA from amyloid-producing E. coli strain and DNA from novel microorganism T. hominis. So conversely, the addition of human eukaryotic DNA had no statistically significant effect on promoting tau aggregation. So next, to compare the magnitude of the effect of different DNAs on their kinetic of aggregation, we measured the lag phase. So the lag phase is just the, it's a time period at which aggregation begins. So the shortest time to reach the lag phase means that the compound or DNA has a high tau misfolding activity. So comparisons of the lag phases indicate that the largest promoting effect or the shorter lag phase was obtained in the presence of DNA from amyloid-producing E. coli strain, non-amyloid-producing E. coli strain, and T. Alzheimeri. As I mentioned before, it's a novel microorganism from the oral cavity of patients with Alzheimer's disease. We then confirmed that the discovered effect of bacterial DNA on tau protein is dose-dependent. So we found that DNA of E. coli at concentrations varying from 1,000 to 10 nanograms significantly accelerated tau aggregation. You can see that DNA of E. coli had a seeding potential comparable to tau seeding aggregates, which were used as a positive control, and meaning that it is really high. So they're doing a very nice job in terms for the triggering protein misfolding. And finally, we performed an electron microscopy to show that in the presence of DNA, indeed, tau filaments are formed. And you can see very nice fibrils that appear following the treatment of DNA, following the treatment of tau with DNA of amyloid-producing E. coli. And under the same conditions, tau alone with the vehicle does not form any aggregates. That is, can be seen on the right-hand side. So we have already published the paper. I think you know that what reviewers were most concerned about. They asked, George, tau is an intracellular protein. and bacterial DNA is not known to be captured by brain cells. So how is your study relevant to what happens in real life? And well, first, pathological tau is characterized by the formation of both intra and extracellular misfolded forms, indicating that the seeding agent could be localized outside the neurons. And the second thing, and it's very interesting, that in brain tissues, E. coli and Porphyromonas gingivalis, they're known to share properties of facultative intracellular parasites and were found inside the neurons. Moreover, some recent data also show the formation of many biofilms inside the neurons by certain bacterial strains. That means when DNA of intracellular localized bacteria is directly released, to the neurons, where tau is normally present, it enables unique possibilities for the interaction of bacterial DNA with tau protein. So, well, finally, the paper was published. Well, although tau protein is an important driver of Alzheimer's disease, the most critical role, definitely, it's attributed to beta amyloid. And to investigate the effect of bacterial eDNA on A beta misfolding and aggregation, we studied the fibrillation kinetics of A beta in the absence of presence of DNA extracted from different bacteria. And sorry, I forgot to make to highlight the names of microorganisms. So let's make clear. So the control was just a vehicle. It was a sterile water. The EC25 is E. coli 80cc2592. It's an amyloid producing strain. PG is Porphyromonas gingivalis. BB is Borrelia. TA, it's T. Alzheimer, a novel strain, and hum is a human DNA. Sorry. We found that DNA of different bacteria taken at a fixed concentration of 1,000 nanogram per mil. Why this concentration? Because according to the literature data, it's one of the highest concentration of DNA that could be found in cerebrospinal fluid or the extracellular part of the brain, components of the brain. So we use the highest concentration, that was discovered. But we did the lower concentrations as well, but just to argue why we specifically use this concentration. And we found the DNA of different bacteria differently affected A beta aggregation. So the maximum pro-aggregation effects were noted for DNA from E. coli, while DNA from other bacteria had not affected A beta misfolding. Sorry, and we're looking right now to the left-hand side. And on the right-hand side, we normalized fibrillation curves to study the lag phase. And well, you can see that any bacteria, even human DNA, shortened the lag phase, meaning that somehow they also triggered or were implicated in A beta protein misfolding. So we can see that the lag phase is shorter on the right-hand side. And the eDNA of E. coli was shown to be the only, so let's start a little bit different. So since you can see on the left-hand side, right? So the eDNA of E. coli was shown to be the only DNA sample to trigger the higher theoflavin fluorescence. So we next evaluated the performance of eDNA from different E. coli species. Sorry, from the E. coli strains. So on the left-hand side, you can see the extracellular DNA that was isolated from different E. coli strains, including those, with from the oral cavity of patients with different diseases. And we used two probes as a control. One of them is a gray one. It's EC25. It's RNA isolated from EC25. So we tried to evaluate whether or not RNA could participate in this process. And the light green or light yellow, the lowest one is EC25 plus DNase. That means it was an extracellular DNA that was treated with DNase in order to see whether or not the oligonucleotides per se could contribute or could not contribute to protein misfolding. So we use clinical isolates, from we need strains. We use different strains and evaluated whether or not they have any pro-aggregational activity. Surprisingly, we found that theoflavin signal was only increased by the eDNA of E. coli 80cc to 592. At the same time, we found that eDNA of all E. coli strains, it shortened somehow the lag phase. I haven't put here directly the lag phase, but you can see that the lag, even on this image, that the lag phase is clearly seen, that the lag phase was shortened. And trying to explain the difference in the aggregation potential of various E. coli DNA samples, we thought that maybe it was due to the particularities of its structure. So we analyzed eDNA sizes using bioanalyzer. So we expected the different E. coli strains could have different fragmentation different fragmentation set of their extracellular DNA. That can be seen on the right-hand side. But we found no differences. We found no differences between eDNA from different E. coli strains in terms of their molecular weight. So this part remains enigmatic, and we don't know why the DNA of some bacteria triggers A beta aggregation and some not. So we can only speculate that there is some maybe specific binding sites along polymeric DNA molecules that are present on DNA of some E. coli strains and are absent on E. coli of other E. coli strains. Even though these eDNA have the same sizes, but we don't have a good answer yet. However, this part is very interesting because our observation that eDNA from different E. coli strains differently affected A beta, meaning that not all bacterial DNA possess equal A beta prenogenic activity, might address the question that I highlighted before of why some people with signs of bacterial presence in the brain develop Alzheimer's disease and some not. So once again, just to reiterate that many papers describe the presence of bacteria or their components in cerebrospinal fluid or postmortem brains, not only of individuals with Alzheimer's disease, but also in samples of mesh controls, even with Porphyromonas gingivalis, yes? And this thing, considering bacterial extracellular DNA as a strain-specific triggering factor of A beta aggregation, might explain this discrepancy, meaning we can assume and speculate that maybe the brains of patients with Alzheimer's disease have strains of certain microorganisms or eDNA, from certain microorganisms that possess this triggering and misfolding potential. Moreover, our data outline a new potential mechanism of how distantly localized bacteria, including those localized in the gut, by the secretion and release of eDNA, could remotely contribute to Alzheimer's disease development and aggravate its progression. Finally, we decided to explore the dose-dependent effect of eDNA of E. coli 80cc to 592. And we added eDNA at concentrations from 0.1 to 10,000 nanogram per mil. So from very, very low to very high. And again, surprisingly, the highest concentration of eDNA at 10,000 nanogram per mil, that is the lowest line you can see, did not increase theoflavin fluorescence, whereas the lower concentration of DNA from 0.1 to 1,000 nanogram per mil had a clear dose-dependent, very nice effect on the beta aggregation. However, please take a look once again, right at this, at this line, at the E. coli of 10,000 nanograms per mil. You can see that it had it shortened the lag phase. It was the most significant factor that shortened the lag phase more than twice. And what does it mean, the shortening of the lag phase of beta amyloid aggregation? Now we're talking about more like proteomic research. So it is known that by the formation of unconventional aggregation. So the unconventional aggregation kinetics could explain actually, this fact, because that means that under the treatment of eDNA at such a high concentration, it leads to the formation of shorter but very strongly, misfolded A beta conformational structures, and that's some sort not of a classical A beta, but it's some sort of intermediate that leading to the so-called off-pathway effect on mature fibril formation. And these intermediates, they're known to possess the highest toxicity. So although there is no clear dependence on the theoflavin fluorescence, we have a very good explanation why it doesn't happen. That happens because such a high concentration of eDNA just triggers the formation of an altered beta amyloid aggregation and altered beta amyloid aggregates. So the next question we'll try to answer, so here we see, dose dependence, and the lower one is a no dose dependence. Sorry, I forgot that I have animation here. So the next question we tried to answer was, is there any effect of extracellular DNA destruction on Alzheimer's disease? And in humans, there is a nuclease enzyme named DNA-1, which cleaves DNA, and well, its function is to clear bodily fluids from cell-free DNA. So we evaluated the effect of gene therapy, AAV-delivered DNA-1 in a mouse model of Alzheimer's disease. For that, we developed AAV-based vector encoding DNA-1 and intravenous injection of the vector with a liver-specific promoter encoding DNA-1 transgene cassette leads to long-term expression of DNA-1 in the liver. And it results in a nice yield of secretable to the serum DNA-1 that also gets to the cerebrospinal fluid. So in other words, we increased serum and cerebrospinal fluid DNA-1 activity and cleaved extracellular DNA in these animals. So we don't have much time and CD in place, so just the results. The decrease of extracellular DNA led to a significant decrease of the microglia activation and reduced beta amyloid and tau proteins depositions that are the main contributors of Alzheimer's disease progression. And and that's it with Alzheimer's disease. And after that, we decided to study if bacterial extracellular DNA could modify other mammalian already synthesized proteins, but non-prionogenic. So we concentrated on effects of eDNA on blood plasma proteins, since they're very convenient for the research, but frankly speaking, actually remain very poorly studied. It was a big surprise for me. I think it was the biggest surprise from 2019, how poorly and how many loopholes are left in the research of human blood proteome. So from the experimental point of view, we used a very straightforward study. Plasma samples were incubated with different DNA probes, then they were boiled, thus the majority of proteins could aggregate, and a fraction of the proteins that were hit-resistant were left in supernatant. And this fraction after filtration was studied with LSMS. The pre-treatment of plasma samples of healthy volunteers, so each set of rows is their DNA of individual healthy volunteer, with bacterial and human DNA altered the composition of the heat-resistant protein fraction. On this heat map, each set of columns is a blood sample, either left untreated or treated with DNA of Pseudomonas aeruginosa, E. coli, staph, or streptococcus, or treated with human DNA. And each row is a name of protein that changed its heat-resistant protein properties in this study. And what we have observed is that microbial DNA has both increased the number of certain heat-resistant proteins and actually led to the formation of those heat-resistant proteins that prior to the treatment were not heat-resistant. So proteins that became de novo heat-resistant are marked with the red circles. I did not highlight all of them, but just to provide you, with the general idea. Moreover, one can see that eDNA from different bacteria had different effects over the same plasma samples. In other words, bacterial DNA induces the formation of heat-resistant proteins. So since prion domains are known to be responsible for protein heat resistance, we next employed the prion prediction plug algorithm to verify the presence of prion-like domains in proteins, which exhibited the changes in their thermal behavior following eDNA treatment. And surprisingly, we found that prion-like domains only in a few proteins which became heat resistant. And yes, here we go. You can see on there only the red box on the left-hand side. All these proteins belong to keratin type 1 and type 2 cytoskeletal proteins. And notably, these keratins were the only proteins which changed their thermal behavior following exposure to human DNA. So human DNA changed only these proteins, only with the prionogenic proteins, and had not altered other proteins. While bacterial DNA changed not only these proteins, but also a bunch variety of other proteins from the blood plasma. And we next analyzed the association between proteins that change their thermal behavior and human diseases. So we conducted a PubMed search and found that the majority of these proteins were previously found to be associated either with the cancer progression or with some congenital diseases. Some of them are even used as a tumor biomarkers. And finally, with that said, we took plasma sets of patients with pancreatic cancer, stage 2 and higher, and compared the profile of heat-resistant proteins of these patients with their heat-resistant profile of normal plasma following the treatment with bacterial DNA. So how to read this heat map? So let's see. The left-hand side, the most left. Sorry, we have here. Yeah, this part that is right now highlighted in a red color. So this is the set of blood plasma of patients with a pancreatic cancer. So each column is an individual sample. So the remaining part is the blood plasma of healthy volunteers treated with different types, treated with different types of DNA. And here we go. So you can see that following the treatment with their extracellular DNA from E. coli or from Pseudomonas aeruginosa, so the heat map just become much more similar to that of pancreatic cancer. And why we have used the DNA of these microorganisms, have not put it here, but there is a number of publications showing the higher rate of presence of specifically of extracellular DNA of Pseudomonas aeruginosa and E. coli in the blood plasma of patients with different types of malignancies. It's an open question, the origin of this DNA, the role of the cDNA. However, it's interesting at least to speculate whether or not it's a consequence or there is some sort of undergoing process of protein misfolding that is specifically triggered by the extracellular DNA of these microorganisms in patients with different types of cancer. So we are currently looking, by the way, for the collaboration to particularly explore this type of study, and the association between the thermoresistant proteins in patients with different types of malignancies. Okay, so yes, here we go. And just to highlight that we don't have a good answer, right, for what does our results mean. Unfortunately, we can speculate that bacterial DNA, by altering thermal behavior of human proteins, could contribute to the malignization process. But once again, prior to showing the role of eDNA as a triggering factor and not a coincidence, much work has to be done. In conclusion, the present studies were to the best of our knowledge, the first outline the universal seeding and modification effects of DNA from different microorganisms on human proteins. We found that DNA from various species of bacteria, some of which were previously identified as a hallmark of Alzheimer's disease, can lead to tau and beta protein misfolding and aggregation. And this suggests the potential role of extracellular DNA in the initiation and progression of pathological abnormalities in different human diseases. So 3 main take-home messages. First is that eDNA from different bacterial species can act as a novel virulence factor and modify a variety of proteins in different ways. This type of modification is not limited to the prion-like templated aggregation. The second one is that bacterial DNA can participate in the cross-kingdom protein misfolding, leading to the aggregation of tau and beta protein. And third, we discovered bacterial DNA as a new therapeutic target for the treatment of certain neurodegenerative diseases. And I would like to thank the Professor Claudio Soto and his team from McGovern Medical School, Casey McGuire from Massachusetts General Hospital, who helped us a lot with vector design, Professor Gabriella from Verona University, who conducted the animal studies, and I would like to answer any questions, or at least to try to do so.
Very well. Thank you for the excellent talk. Yeah, in a few minutes, you open the discussion. I actually wanted to ask you more like a general question, because so you and Victor